soldier, because he's the one who's going to have like kind of survival of the fittest. So every cancer cell is kind of a like a Indian soldier, where he will try to like uh, fit into the environments and conditions, and what not things that will happen during the war sessions or something like that. So I would see cancer very similar properties of a military person, and they have their own, like how soldiers have their own characteristics. Similarly, cancer also has a lot of hallmarks. So there are different hallmarks, especially uh, there are like 11 hallmarks in cancers. These are the major parameters that expresses saying that uh, these cancers will survive, and these properties will help them to be there and grow themselves in their region, and then start getting like proliferated and differentiated based on the genetic factors, and also like inflammation that's creating and resisting to their death, and what kind of regulating pathways that are happening. So they also try to modify themselves into a new kind of mechanism. That's where they still stay in our body for a pretty long time. So rather than cursing the cancer, I would also like embrace cancer, because the way that these cancer cells are trying to convert themselves into a like a kind of a soldier, or they are trying to embrace themselves in their in a harsh environment, like say body, because we have our immune system that's going to like you know going to fight with these cancers. So I will always embrace cancer. That's how I love problem to solve this. And basically we have these hallmarks of cancers can be basically used for I'm going to look more on the monitoring the disease progression, because when we think about a drug that is developed from a bench to preclinical testing and then moving to the clinical testing, it's going to have a long process. And during this process, it is quite important to have two important points. One is the monitoring of the disease progression, how well you can able to understand these cancers are getting survived. And second one is, if you are giving any kind of drugs to the patients, and how these therapeutic response is going to be there with the cancer when the drug is interacting with these cells. So these are the two major points that we would like to understand during the process of drug and also the general the cancer progression, right? So based on this, we wanted to develop different contrast agents and markers. That's where there are different contrast agents that helps us to differentiate an abnormal to normal cells, right? So it's a kind of a medium, or I would say it's an exogenous material or a synthetic materials that can be used for detecting structures like say a cancer when compared to a normal cancer cells. So here is just one image that represents a brain MRI with CT contrast, or sorry, with MR contrast and without an MR contrast. So we can see the differentiation between where is the localization of these cancer cells behave. So that's where these contrast agents are more important. As we see, FDA has approved many kinds of contrast agents for different kinds of imaging modalities. If you see on the right side, you have a lot of molecular imaging techniques. You have a molecular imaging techniques, and also you have anatomical imaging techniques. So if we talk about anatomical, majorly focuses on X-ray or micro CT or a CT imaging systems, and we have MR imaging systems and ultrasound imaging systems. And apart from this, if you want to know the dynamics of how these like cancers work, so then we try to move towards more on the radionuclide imaging and also an optical imaging. So, but not always these individual technologies are helpful, but in current trend we need to have a combination of these two technologies together. Like say for instance, we wanted to, if you look at in a general clinic in India, we have both a CT and a PET together. So it's not for cancer imaging. We always use a combinational imaging systems, which can be a PET CT or PET MRI. Sometimes people use CT ultrasound, sorry, ultrasound optical imaging, and MR optical imaging. So there are different ways of technologies that are coming up with. So hybrid imaging modality is one of the key factors for understanding these cancer-based diagnosis and also monitoring the treatment response. So the biggest problem with these contrast agents is that the two major things that I would suggest. One is how well these contrast agents, like say these FDA approved based contrast agents, are clearing mechanism in the body, and also if you're talking about any kind of like say dye or looking at in a cellular based imaging, then we use more on degradation. So either two things can happen. One is it can clear faster in the body, and it will degrade due to the excess amount of photon that's interacting with dye. So these are the two major things, but these are the limitations where we need a sophisticated contrast agents that can be useful for cancer imaging applications. So there are two different okay. So cancer diagnostics, I would define as where using these kinds of contrast agents, both can be used in bioimaging applications and also for therapy. So that's where we can have nanomaterials in current clinical health, mostly focusing on the drug delivery applications, tissue engineering. Right now, tissue engineering is going much more faster on what we are trying to see on 3D bioprintings. So people are coming up with 3D bioprinting and 4D bioprinting. People are coming up with shape polymer alloys. So even though these materials are useful, somewhere these contrast agents will also help them to create and mimic structures that helps them to create a proper structure for it, right? And also the other part is like the diagnostics. That's the next revolution that we are looking at, where currently all of these point of care testing that comes under biodiagnostics, where contrast agents is used in these kinds of technologies. But the sensing mechanisms can be different, but again, you need to have some kind of signals that are generated when you're looking at in live, or you wanted to have some kind of coloring dye that is used, like say for instance gold nanoparticles are used for like say Prega kit test. We have like immunobodies, antibodies are there. So even for that, we need to have some kind of diagnostic devices, and nanomaterials really helpful for that. And apart from this, we have a combination of fields that are required. One is nanotechnology, imaging, and microfluidics. This is how my whole topic is going to focus on. So how these nanomaterials is going to help in these applications, and also what kind of fields that's going to use for making these materials, and also how we can use that materials for imaging, and how microfluidics is going to helpful in tissue engineering, biodiagnostics, and drug delivery applications. So there is a lot of uh, I mean, like a bridge that happens between the clinical health versus these fusion of fields. So it's very important to have combining this fusion of fields and the clinical health, which is going to speed up the clinical translation. So that's one of the key point that's very currently lacking in today's regime, because a drug is developed, but it is not properly delivered inside the body. In that case, we need to have some kind of nanomaterials to speed up the translation process. Right. So one of the things that we are going to look at today is the first part is going to be more on the colloidal gold, or I would say the gold nanoparticles. So these gold nanoparticles are can be divided into like the three different shapes. I would say like three different uh, like types. So one is I would call it as clusters. So clusters is the smaller size of a general nanoparticle that we synthesize in the labs, which is going to be like 10 nanometers. But the clusters are going to be a few atoms that's that's agglomerated together to provide some kind of structure. So typically it's going to be in like 1 nanometers, less than 1 nanometers, 1 to 2 nanometers. But the general nanoparticles that we see is going to be like a reddish pink or a wine red color. So those particles are going to be in a spherical, which is like 5 to 10 nanometers, typically more than 10 nanometers. That's what I have synthesized till now. So, and apart from this, we have some anisotropic structures which has different sizes and there are different shapes, and these kinds of particles are divided. So these colloidal agents are used for a variety of applications, especially if you look at these clusters. These clusters are fluorous in nature, so we can use these materials for not only as a therapeutic applications and also for imaging application. We can use these for like say hypothermia. So that's what the therapeutic applications is. But spherical nanoparticles we talk about more on the X-ray imaging, but anisotropic can be used for like say sensing applications. Right now Dr. Gauri, I think she's working more on the SERS based sensing. I hope she has a lot of experience in and creating a different kind of structures of these gold nanoparticles and using it for SERS sensing applications. So after this, if you look at the different applications, we need to think about a few properties. One is we need to make these gold nanoparticles to be more tunable, and also we want to make it more like homogenized. I mean, like homogeneous structures in size. I mean, like morphologically all the nanoparticles have to be in the same size, and it should be like functionalized, and it is capable of getting functionalizable. So materials are more tunable, and also they are more functionalizable. It can easily functionalize. That's where people are more prone to use these gold nanoparticles, and a lot of papers have been explored these applications. And another important aspect of gold nanoparticles is that gold is mostly a biocompatible materials, wherein it's not going to cause much of toxicity to the in vivo region, and also for like say in clinical applications. But it is not completely moved to a clinical side, clinical sector. But right now it is still in the in vivo, and over the time it might use it for like clinical applications as well. But the biggest problem here is the synthesis of these colloidal golds at a high concentration is one of the biggest problem. So that is where it lacks in using it for clinic. But in preclinical applications they are most suitable, and we can able to identify the drug analysis and also like the interaction of drug and how these drug are like distributing throughout the body, right? So and another important point where we are talking about more on imaging is that we were using these gold nanoparticles for X-ray attenuation, because gold nanoparticle is a high atomic number. So that is where these gold nanoparticles with high atomic number has more absorption of X-rays compared to a general tissues, right? So these gold nanoparticles has a very similar absorption characteristics like a bone in our body, right? Sometimes it is like more than and a little bit more than the bone's attenuation factors. That's why people use gold as a prominent contrast agents for like bioimaging applications, right? So the other one is that we are talking about more on the RF hypothermia, where interaction of these nanoparticles can be more on the gold, where it absorbs these nanomaterials, these electromagnetic radiation such as radio frequency at different megahertz of frequency, and that is exposed to these gold nanoparticles can also help in therapeutic application. So we could able to see the gold as more tunable, functionalizable, and biocompatible. And not only that, it's more related to the therapeutic application and also for an X-ray imaging application. So one such work, what we try to do is that we wanted to develop a gold nano particles with different sizes, and we wanted to test it in our in vivo for heart vasculature imaging applications. Uh, So the one physical synthesis that we use is a simple citrate-based method. So we are able to develop two different types of nanomaterials, which is like 2 nanometer and 10 nanometer particles. And these particles are used for bioimaging application. Especially, we wanted to look at how these nanoparticles, when injected in tail vein, what is happening to these kinds of understanding the hard dynamics of mice. That's one of the critical problems that we were working on. uh, so we understood its characteristics, like say optical properties, and we try to use these optical properties for both like imaging applications. That's where, if you see on the on this image that's there inside, so the fluorescent, it says clusters of particles, which is like gold nanoclusters, which is conjugated with like BSA, that's a bovine serum albumin. So if you use a simple strategy of using these gold chloride and adding it to a like say bovine serum albumin, or even you wanted to use something called glutathione. That's another chemical that can be used. And apart from this, there is cyclodexane, so you can also use those chemicals. And these are the different like functionalizing agents and also reducing agents, which helps in the formation of gold nanoclusters, right? So the clusters is going to be in the range of, like say, the number of atoms that are combined together. So it's going to be more than like 25 atoms to 70 atoms, depending on how these like particles are interconnected, right? So the other one is that in this project we use more on the glutathione. So that's why you could see the GSH based GNCs, but you can also use bovine serum albumin. That's also one of the easy available uh, like agent. We can use it for making this gold nanoclusters, which will give a bright fluorescence when you excite with UV region. So this is very much helpful for like biosensing applications like cellular imaging or cellular dynamics that we wanted to look. And it's also good fluorescent properties, than very equivalent to a like quantum dots that are available. But I wouldn't say that it's the intensity is far more better for quantum dots when compared to the glow clusters. But these clusters are more biocompatible compared to a cadmium selenide or cadmium telluride, which has a more biotoxicity. But these you know, clusters can be used for imaging applications. So during our time when we were trying to develop these microcity system, which is an in-house built microcity system in IIT Madras. So we use these kind of like nanoparticles for imaging applications. So we built our own microcity system in the lab. So if you can see, a general CT works based on the rotational motion of uh, 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 X-ray source and the detector around the patient. But here, the rotary stage, we have a rotary stage in the center, right? And we have an X-ray source that comes from here and interacts in the mice restrainer, and you could see the X-ray attenuation that hits on the scintillator. So this is a scintillator where it converts an X-ray into a visible photons. That's what the scintillation mechanism is. And from there, you have a 45-degree angle mirror that helps you to divert all the information from here to the camera. So we came up with a low-cost microcity build system, which is covered completely with a lead sheet, because the X-rays are more dangerous. So we were trying to use a surface coating mechanism on the sheets, which is basically on the lead phenomenon. Right. So this is a projection image of this image when we have a small sheet which has different sizes of these dimensions, and we were trying to image it in this. And we used a mice restrainer because our aim of the project is basically to see how a nanomaterials can go through heart vasculatures for a mice imaging application. So that's the one of the key challenge here, because uh, uh, normally contrast agents are used for the general animals, like say rats. But when because when you're trying to use these contrasts for rats, it's just going to be like say 100 beats per minute or 100 to 150 beats per minute. But when you're trying to go for uh, mice, so what happens is you have much more higher heart rates. So that goes up to like say 450 beats per minute, 300 to 450 beats per minute. So in that case, we wanted to image. We need to have a very fast scan rate of imaging this heart mice, right? So that's the major problem that was encountered during the usage of these materials. So then what we thought is, let's try with a simple iodine injection, and then we wanted to try this with our inbuilt house microcity system. So what we did is that we used a cardiovascular imaging, especially to see uh, how these iodine based agents, which is going to which has a very similar characteristics of gold nanoparticles, but again it clears faster. That's the one of the biggest problem, but still wanted to see how this imaging can be done. The advantage of using this is that uh, doing a high fast frame rate camera imaging for X-ray is much tougher when compared to a general X-ray imaging, because you always form a kind of a shadow, but you need to go at least like 40 frames per second, which is not possible with the current technology that we have to visualize the mice cardiac imaging. Right. So what we did is that we did a 40 frames per second with a 50 kilovolt X-ray voltage of 1 milliampere of current, which is used for imaging carotid artery intravena cava, where we did a tail vein injection. We started injecting through the tail, and it starts reaching through the IVC, and then it reaches the coronary artery, right? And from there, it started reaching to the lungs, and we have like pulmonary artery, and we can able to see how the aorta, the thickest vein that is forming between the region, right? So this is how we could able to see a strong, like uh, long-term imaging for these like cardiovascular diseases. So this will really helpful. This imaging modality is going to helpful to understand if there is any kind of cardiac stent based systems that can be measured, or if there is any other problems related to the heart. So then this kind of imaging systems are required. So in this case, we also need to know this imaging agents are going to be play a major role for doing coronary artery. And it was really a challenging task for us to do cardiovascular imaging, especially for these contrast agents. And we also did same for a corridal gold, but we were not successful in doing it for the clinic, because the injection did not do well, and also it started it was much quicker, and it was not easier as a iodine based contrast. But still we could able to see some kind of contrast that is being stored in the vascular region, and also in the respiratory lungs region. So, and also we thought about all these contrast agents are really helpful in molecular imaging, because as I said, X-rays can not only use for a general imaging applications, but we also had a question whether X-rays can do a molecular imaging, because structural imaging is possible. It's been known since 1800s, but can we do a molecular imaging using an X-rays? And people have understood that molecular imaging is possible by using the same scintillation mechanism, like how we were talking about X-rays can be converted into like light. So we can able to create some kind of a scintillating materials, and we can use these scintillating materials, which is more biocompatible, and we can able to use this for imaging applications that can also be used as a molecular imaging. And not only molecular imaging, because this material is going to also have a fundamental aspects of contrast agents like X-ray CT contrast, so we can also use as a multimodal imaging. So the basic principle of this molecular imaging is that when you apply an X-ray on a contrast agent, what happens is it goes to an excited state. It basically ejects a deep electrons, and deep inside the electrons, and those electrons are excited. When it comes back, it gives luminescence from the material. So you have an X-ray agent, and you apply an X-ray where you get luminescence from this material. So one example is that if you see on the right side, you can see a clear distinguish between UV excited fluorescence versus X-ray excited luminescence. So that's what I was mentioning about how the scintillating mechanism works. So if we are developing a material which is more biocompatible, and where if this can be applied in ejecting the core shell of electrons to the conduction band and then reaching to the valence band, thereby it can provide some kind of luminescence. But the important aspect of why people wanted to make use of this is because UV or light based imaging is much more complicated, because you have a lot of background fluorescence. But the same image, if you're trying to look at in an X-ray luminescence, then you have a very much small background imaging which you can completely avoid when compared to a general UV light based or a general light based imaging. So that's where people are more excited to develop these kinds of contrasts, and it's very important to have an X-ray luminescent imaging which can be used for both X-ray CT and also for optical imaging application. So one such idea is that we came up with to develop some kind of contrast that can be both visualized in scintillator, where if you see once the contrast agents are injected in animal, so you can have an X-ray source that can be visualized in scintillator, and those materials can also emit a luminescence. So that's kind of a dual properties that you can get from a single imaging agent. Then we can able to do a complementary image mapping between XCCT and XEOL application. So this reduces optical autofluorescence. That's one of the key problem that's there with the conventional systems. And again, you have a penetration power of this X-ray is much more higher. And the biggest thing is, you have a high resolution image. But one key problem with this technology is that X-rays are like more radiation based. That's one of the key problem here. But then we can use these X-ray contrast agents for both dual applications. But if you are not looking at on the X-ray source for radiation, but if you're using for a short term, then this technology is going to be perfectly good for complementary imaging application. So the one such combination which has both of these technologies, that we came up with a wide known material which is called the tantalum. So tantalum has a very similar attenuation properties that we could able to see in a gold, but tantalum oxide is much more a biocompatible material than than the gold nanoparticles. So one thing, if you see here, is we have trying to dope with europium, because europium has a property of Zeol mechanism, but when it's like combined or synergetically combined with the host mechanism, which helps to provide a dual emission. So as we know that tantalum oxide is a very good X-ray contrast similar to gold, but we wanted to see whether europium is going to fit into the tantalum oxide nanomaterials, and thereby it can provide some kind of sense for imaging application. So what is our thought process is to see we can make a dual modal imaging or a single contrast agent that can be visualized in both X-ray imaging and also a CT imaging application. So this material is a wonderful material. Then we started to expose these materials with X-ray with different properties, and we could able to see a beautiful orange contrast when exposed with an X-ray region. So that's one of the biggest thing that we could able to do, and this is currently patented, and we are trying to sell this technology to the companies where which can be used for contrast for GE systems, because like the other biggest problem that we currently face is that the translation possibilities or problems that we could see where the hybrid imaging technologies are not completely used in research based or even in the clinic based, but still this is one of the example of contrast where we can able to show that it can be widely used for different applications. And the interesting point here is that people haven't noticed that tantalum oxide also could see a little blue emission. That is where our overall mechanism of emission changes when you have dual emission properties. So as I said, you have an optical luminescence when you have an X-ray contrast, but also you have an emission properties in optical region. So you have this powder. So when you excite with UV, you also get a luminescence. So once you take some kind of tissues from in vivo, you can able to image it in cells or microscopes. Also, you can use it in vivo directly. So those are the three advantages that we could able to use these X-rays for imaging applications. But can we use these X-rays for knee regions? So that's what we wanted to see here, because where all these X-rays can be used, where X-rays, especially we are looking at in the cartilage regions, and cartilage is much more tedious, a tissue which does not have any blood vasculature. So when you're talking about a general knee osteoarthritis condition, a lot of people who have this problem with normal and abnormal knees, and it's going to be increasing. The global population of more than 40s is going to have a prone condition to a knee osteoarthritis because of these factors and pathophysiological condition, especially the systematic risk and local risk that combines together with the pathophysiological condition, which is going to like have ROS induced local articular lesion and also an synovial inflammation. So the basic diagnosis happens at different modalities, but the X-ray CT is one of the primary modality that is used in clinic, right? So we can able to both use for OA grading applications, and also we can use this for molecular imaging applications. So what we wanted to do is that we wanted to see if there are any contrast that would typically help us to visualize the penetration of these nanoparticles into the cartilage. So what we had in our mind is that to come up with some kind of nanomaterials which should have particles less than 50 nanometers, and we should also see whether it is modified with surface, and also it should have a positively charged particles, and they should also have a biocompatible material. So when you're looking at the cartilage as a whole, to visualize, to see how these knee osteoarthritis are getting degraded, or especially we're looking at the cartilage degradation. So then we need to look at a interesting contrast that we needed. But the widely used contrast, which is called the cerium oxide nanoparticles, because previously we have used tantalum oxide, but now we are trying to focus more on a ceria nanoparticles, and we wanted to see whether we could able to get the same properties like how we have seen for tantalum oxide. So this has a dual applications, both because ceria is a very good X-ray contrast, and also we wanted to do with europium to see whether these materials can be helpful for osteoarthritis imaging. And the ceria nanoparticles are more positive. In that case, we can able to like interaction of these materials to knee osteoarthritis is going to be literally high. Right. So we started to synthesize this with a simple combustion-based technique, and we were able to get a monodispersed nanoparticle structures with a size distribution of like 50 nanometers. So we're able to generate particles which less than 20 nanometer, which means that these particles are going to penetrate inside the cartilages. Right, so we did some kind of a testing. We created a small phantom model images, and we loaded these images with these phantom models with different kinds of cerium particles, and also europium doped cerium oxide nanoparticles at different concentrations. And we want to test it how these UV excited emission that's coming up with. But the biggest problem that we find here is, though the host material of tantalum oxide was much more sufficient for providing an X-ray attenuation, but cerium did not provide us a host material for giving an X-ray luminescence. That was the biggest challenge. So we tried understanding the properties, but then we failed to get X-ray excited optical luminescence with these technology. So what we are trying to say here is the host material is much important when we are trying to look at an X-ray excited optical luminescence imaging applications. So, but then we try to understand more of these contrast for X-ray knee osteoarthritis condition, basically the cartilage degradation. And also, we are looking at more on the contrast properties. So we did a contrast for dual imaging application. So we did a controlled materials, which is like a 10% europium doped gadolinium. So gadolinium is a X-ray contrast again. Europium is a dopant material which is used for as a positive control, and so this is combined with connected with like uh, like differentiating between the water control and also different concentrations of cerium oxide and europium doped cerium oxide. So the numbers that you see here is 10 to EC is basically E is europium based cerium, and C is just cerium, and you have the different concentrations of europium 2% and 10% with the different heating parameters of like cerium oxide nanoparticles to form the structures. So we wanted to try this with a goat cartilage imaging. That's where our intention is, because the one thing is how these nanoparticles are engaging with these like cartilage applications. So what we did is that we got a cartilage from the local butcher shop, and we started to do a small biopsy punches of these tissues, and we started harvesting this with a controlled nanoparticles and non-controlled nanoparticles, depending on what kind of materials that we are trying to focus on. So based on this, we could see an optical imaging which has a confirmed luminescence dependent on the dose-based materials. So this indicates that the accumulation of nanoparticles is higher. And again, quantitative analysis showed that the uptake up to like 10 mg per ml with no difference between 1 and 5 mg. So 1 mg is much more better, and 5 mg is also good enough to do a imaging application. So we also, once we do this, we also tested with a simple staining technique with HND. So we use the cerium oxide nanoparticles for superficial region and also for safran and O, because safran and O stains the glucose aminoglycans. That's the major polymer that we could see within the protein matrix that we could see in the cartilage region. So we could able to see the penetration of these cerium based nanoparticles that is especially visualized in the optical imaging applications. So we also did the same thing for different applications, both with and without nanoparticles. We could find the safran and O conditions are slowly changing, and thereby the concentration is impacting a major role in the degradation of this glucose aminoglycans. And apart from this, we also started to work more on the theranostic approach, because we were working more on the nanoparticles for immunization, but we wanted to see how these materials would work for theranostic applications. So one such application that we wanted to look at is that how these nanoparticles interact with RF, because it's going to be a good theranostic condition, and Kanzius was the first person who's provide an interaction of gold nanoparticles with RF interaction that produces a heat. So we have some questions when we try to work on this RF mechanism, where whether the nanomaterials can be heated, or it's not heating, it's just because of the condition that's applied on what mechanism these nanoparticles are heated, and whether we have some kind of in vitro or in vivo cell death that's happening, is basically some kind of a biological effect that causes a therapeutic effect. So on what basis we can able to conclude that these colloidal gold nanoparticles are heating when exposed with RF? So we were so excited to see. Then what we planned to do is that we started to have these two nanomaterials, and we started to come up with different frequencies, exposing these nanoparticles at different frequencies, and we wanted to have different power levels. So varying the parameters of frequency and the power levels, which really is going to helpful to see how these nanoparticles will behave, because over the decades people are still believing that the gold nanoparticle is going to provide heat when you apply an RF energy. But the interesting factor, what we try to do is that we wanted to understand more detailed mechanism of these, contrasts, because as we use this as an X-ray contrast, which can also be used as a RF, because we can try to combine the modalities of X-ray and RF together, so that we can have a like synergetic theranostic probes. So as an hypothermia setup, we started to come up with like a simple 27 megahertz with a tissue phantom, and then we wanted to study its heating and cooling rates. So what we did is that we wanted to use a simple PBS that's going to mimic a biological substance. And again, we created three different holes, and these holes will be loaded with some kind of nanoparticles between 2 and 10 nanometers, and we want to see how these like frequencies are going to help us in doing it. So the top one is basically used a phantom RF antenna that is placed at the bottom, but here we place the antenna on the right and left. So on both of these parameters we were looking at the frequency of based on the different frequencies like 434, 915, and 27. We want to see the heating rate and cooling rate. So we wanted to compare this with the microwaves and also radio frequency to see the heating and cooling effects. So what is the final result is that we found that there is no heating that happens when you try to heat with radio frequency. So both with 10 nanometers and 2 nanometers, different powers and different concentrations and different frequency levels, and everything confirmed that there is no change in the heating rate. But there is a little change because we're looking at a more concentrated, but the final, the delta T varies is very minimal, which is like almost equal. So we came to a conclusion stating that the 3 point, like these gold nanoparticles does not heat when you apply an RF. So we started to work on more on the fluorescent based mechanism where iron is one of the key parameters, because we've also realized that hypothermia can also be used with iron nanoparticles, so wherein RF can be exposed with iron. But we also try to understand how this iron would help us in RF imaging applications and RF hypothermia. So we also found that iron can be used for both an imaging application and also for an hypothermia application. But we were more interested on the treatment purpose of using these imageable nanoparticles, because iron is used in MRI, which is widely known, but we wanted to see how these materials can used as an optical imaging, and also how we can use these iron as a therapeutic application. So we started to develop an iron quantum clusters, which can be used for both imaging agent and also for a therapeutic application. So we started to evaluate ion quantum clusters of both these things. So this one of the uh, research my student who did it, and she said the UV based fluorescence that's coming up when you excite with the UV, and also we have some kind of heating effects when you expose with radio frequency based therapy. So here we wanted to try this with quantum clusters, because the cartilage regions has a higher, variations that can be used for, like improving the cartilage factors. So that's where we were trying to use these ion quantum clusters as an agent for so our important understanding is that if we have these nanomaterials and quantum clusters that can pass through a cartilage imaging through a superficial layer, and we have extracellular matrix, and we can use this for tracking how the regeneration is happening. So this basically for understanding the bio tissue bio imaging applications. So we try to use these clusters which can penetrate inside the cartilage, and later if there is any change or improvement that happens within the cartilage, then we can able to visualize that. So it's basically for track the regeneration imaging, and also these imaging agents can also be used as a hypothermia applications. So we wanted to test both of these. So we also wanted to see how this quantum clusters are different at the same frequency of 27 megahertz, because already it is proved with 13 megahertz. We want to try with the 27 megahertz. So we used piperidine and the hemoglobin. So these are the two things that we have used for making these materials. And when we use HA, because hyaluronic acid is one of the key biopolymer that can be interact with the cartilage, and uptake can happen much more easier way. So we use the thermal imaging cameras for understanding the diagnostic imaging. So we did a whole screening of plain HA, ion quantum clusters, and HA quantum cluster screening. So we could not able to see much of difference here. Again, there is a slight variation that's happening. You can see a large statistical differences between these levels. Because what we are trying to say is that it is not being the heating mechanism is not uniform, so there is some kind of changes that's happening at each and every stages. So for HA and FE quantum clusters, we could see a large the standard deviation. So this is due to the improper heating effects that's happening with the quartz clusters. But still, we conclude that ion quantum clusters do not heat, even though we have some kind of HA. But HA is kind of a little bit more on heating effects at lower concentration. We are still not confirmed on why this HA screening is heating up. It could be because of the problem that arises with some kind of ions that is dispersed in the material or the water. So that could cause some kind of heating mechanisms. But overall, quantum clusters do not heat when you end up in like understanding these clusters. So we also tried this same thing with the cartilages with the biopsy based, and we found that the ion clusters have entered inside the particles, and we can the cartilage, and we can able to see when it is excited using an UV. So similarly, we also tested at different HA concentrations and also different sizes of HA, the different concentrations. So both of this able to see show that when radio frequency is exposed with these kinds of concentration of ion clusters, which could have a drastic amount of change in the heating effects. So ion quantum clusters and HA together showing in heating effects, but also the simple ion quantum clusters is also showing it. So there is a lot of like the results are not that clear for us to understand or conclude what kind of heating effects are coming up with ion clusters. So next is we want to look at more on the molecular imaging. So we started to use a quantum dots for a variety of applications, like say quantum dots for imaging, optical imaging, and the microscopic imaging. So one such quantum dots that we wanted to use is that graphene-based quantum dots, which is widely known for different applications and also for different properties, and where we synthesize one, graphene-based quantum dots using a laser ablation technique. And what we arrived with the laser ablation technique is that we could able to visualize these quantum dots for both near red quantum dots and color tunable quantum dots. So you have wherever you excite. So the color tunable is that when you excited UV, you get a green emission. When you excited green, you get a red emission. So depending on how you wanted to, tune the properties of these quantum dots. Right, so we tested basically oxygenated graphene quantum dots, which means that we have not added any kind of uh, dopant agents to give a fluorescent agent for coloring application. So we developed these quantum dots with the size of like say 5 to 10 nanometers that is dispersed. Probably like more 2 to 10 because 2 nanometers are more blue in region and 10 nanometers are red in region. So we could able to see a wide range of fluorescent properties that comes up with two heating ways that we look at. So here, if you see at different excitations here, we have different emission properties, and thereby here we could see a color tunable emission which is focused on the different excitation properties. But the comparing both of these is that on the overall, the average peak ratio is mostly on the 600 nanometers, which means wherever you excite, the emission comes at 600 nanometer, the maximum peak that's coming at 600. But when you're looking at in the intensity based one, so it shows at a different color tunable, but also different emission properties. So this is much important. So that's how we understood that these quantum dots suggest that the electron transition happened from HOMO 1 to HOMO ground to the LUMO state. So that's where the transition that's happening for having a color tunable and also a mono fluorescent based nanoparticles. So we also tested these are results, which shows that the particles with 20 and 1 is the two heating ratios that we see here. So at different excitation, you can see the different color emissions, wherein we have all this emission filters and excitation filters because we have different LED sources, and also we have different emission filters to see whether we are looking at a proper, uh, specific region of field of interest. So we can also see the quantum dots for cell imaging, and also we use these for long-term imaging applications for like say 48 hours to 72 hours. But we have some results with 72 hours, and also we see how these nanoparticles are interacting within the cells. So if you see here, the cells are mostly are on the both on the top surface on the cell, and also it is also there in the cytoplasm. So which means that the particles interact more on the surface, but it penetrates so faster during the time, like say 24 to 48 hours. So even at 48 hours, we could see that the particles are still sitting inside the cells, and it does not cause any kind of toxicity. So that's one of the key factor that we could able to see that without any doping agents, we could able to develop a color tunable quantum dots, and also red emission quantum dots, which can be used for bioimaging applications. And we want to test the same thing with in vivo imaging applications. We created a small gel loaded with these quantum dots. We want to test it at like a trans illumination based imaging. So we used a filter of 590 nanometers and 16 nanometers. We want to see whether there is any luminescence imaging. And we did a merged imaging of both optical imaging and fluorescence. We could see that the position that is there in the region shows a better optical imaging application. So this is much more better for the general imaging. Uh, so we have a nanostructure synthesis for microfluidic applications. This is the last part of our topic, like how we can see microfluidics can also be used as a basic reactors for nanostructures. So we need to have a large facility to develop these kinds of nanoparticles. But can we fit into a small chip? Can we fit a whole lab to a small chip? Whether is it possible to do that? But let's look at some you know differences between the batch versus the lab on chip. So the batch reactors changes by batch, wherein the lab on chip mostly works on the continuous base synthesis, and they are highly reproducible. And we have a negligible waste when compared to a general batch reactors, because if you are trying to conjugate some kind of dyes or drugs onto the contrast agents, then you need to have a large amount of dyes, right? And we also the whole process changes depending on the reaction. Here depends on the design of the chip, so how you wanted to make these nanoparticles. So this is very much important for translating from a bulk to a lab on chip technology. So understanding the microfluidics from the nature is that we can think about xylem and phloem, where the water and the food is transporting one way and two ways. Similarly, the microfluidics is gonna work on the same way. But if you look at on the river, that's also can be converted into a laminar flow and turbulence flow, where if there is no change, then the liquids can go pass directly. But when you have a turbulence like a small hum, then you can see some kind of change in your fluid flow reaction. So the basic understanding of these nanoparticles is that the formation of nucleation growth and the growth over the time that happens from nucleation, aggregation, and then slower growth and then faster growth. So this is the point where it's going to change its size of the particle, and whenever you wanted to stop, you bring it out and then and start storing it, right? So we started to work on a simple lab on chip based device to develop contrast agents for like bioimaging applications. But we initially wanted to try to make a different structures of nanoparticles. That's one of the key factor for sensing applications. And also the one application that we are trying to do here is for loading drugs inside the cell. So we used different kind of different structural properties of different structural sizes of gold nanoparticles to see if we can able to use that for optoporation applications where you porate the cells and then you deliver the drugs inside the cells. So we started to develop two different types of nanoparticles. One is a bone-shaped structure, another is a spherical shaped structure. So which is basically kind of 30 to 35 nanometers in size, and this one is the long is like say 60 nanometers with 20 nanometers in the width. So we did this using the design that we created here. So we have an ascorbic acid, a growth solution, and a seed solution. So this forms a droplet in this region, and it helps us to incubate and then mix, and then slowly it forms the gold nanoparticles through the solution. So the combination, the flow rates is all important when we try to create these kinds of nanoparticles for imaging applications and sensing. So finally, we did a calcium AM loading into the cells. So with and without these nanoparticles, we did a small staining applications and see that we could able to image whether the cells are alive when we are interacting with an optoporation of these say gold nanoparticles and bone shaped structures. So and to quick conclusion, we have this contrast agents for imaging and therapeutic applications, and also for 2D and 3D structural imaging. And they could see a large amount of interaction of X-ray excited optical luminescence and graphene quantum dots. And we can also use this for large scale synthesis of contrast agents. Yeah. And I would thank the organizers of the refresher course, Dr. Gauri, Dr. Uttam, and Dr. Kalpana. And thank you so much for inviting me. And I'll also thank my mentors here, Dr. Jai Kumar, Dr. Ganapati from IT Madras, and Dr. Tuhin from IT Madras, who helped me doing a lot of research. And thank you. Open to questions. So if anybody has any questions.